This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 495, recorded on May 25th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, where it is 30 Celsius, and mostly sunny, Dickson, Despommier.
Good afternoon, Vincent.
This is our third podcast of the week. My fourth.
That's right.
Dickson and I spent 3 hours in a car together this week.
One per day.
Boy. Good thing we weren't podcasting, so we got along, right? Also joining me from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
All right. All right.
We got 90 degrees here Fahrenheit. That's 32 Celsius and a mostly blue sky, a couple of puffy white clouds. We're headed for summer here. In a week, forecast is to be in the breaking 100. So
wow.
There you go.
Went from cold spring to intolerable summer.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. I'm excited to tell you it's 82 degrees Fahrenheit, so it's 28 Celsius. And the sky is completely blue and not a cloud, and it's gorgeous.
Right.
So I suppose in the summer, it's every day gets to that, right? Because it goes over 82.
It gets hotter. Yeah, it does get hotter.
Do most days in the summer go over 82 in Ann Arbor?
Somewhere around there. Sometimes we're in the 70s. Sometimes we're in the 90s. It just varies.
I bet in Austin, it's always over 82.
In the summer, yeah, probably. I'll keep better track of it this year.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 87 Fahrenheit here, 31 C.
You probably don't have 80 every day in the summer, right?
No, it varies between the 60s and the 90s depending on weather patterns.
I mean, we have the ocean to keep us cool until it warms up.
The microbiology department at Icahn School of Medicine is seeking faculty candidates interested in strengthening their program in virus host interactions. Applicants must hold a PhD and or MD, have postdoctoral experience, and be interested in creating a virus-related research group that will complement the pre-existing departmental program. Recruitment of individuals or couples at the level of assistant, associate, or full professor will be considered. More information about the department and the school can be found at icon.mssm.edu. If you want to apply, send your CV and a brief description of your research to nycvirology@gmail.com, or you can contact Ben Tenhoover directly. ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, June 7th to 11th in Atlanta using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial sciences in the world. Delve into your scientific niche in 8 different tracks. Don't miss this opportunity. Visit asm.org/microbe, asm.org/microbe, and use promo code ASMPOD for $50 off registration. See you in Atlanta.
I also know that the mobile app for ASM is supposed to be released on the 29th of May, in case anybody was anxiously looking for it, like I was.
Any news from ASV?
ASV is going to be really big. 1,401 people are registered as of today.
Goodness.
Wow.
Yes. And so there's a little pressure on the hotels that we had room blocks for, but University of Maryland has opened up 100 extra singles for on-campus housing. And then there's plenty of hotels in the area not in the room block. In that whole area, just oodles of rooms. So you should be registered by now because now it costs more, but get your housing squared away if you haven't yet.
So this must be a record.
No, turns out that we have records back to at least 2001, and there were 1,600 in Madison that year.
Ooh.
Oh,
Yeah.
okay.
Actually, our records go back to 1994 in this document from Ann Palmenberg. But yeah, so 1,600 is the record. We have a ways to go.
Does anyone know how many virologists there are?
I don't know, but I did have to try and check whether ASV was the largest virology society. And depending on how you count the numbers, the European one might have more, or we might have more.
When you do express it, though, it's 10 to the
30th.
Right.
Right.
If you line them up end to end, how far would they go?
But both of those have memberships. Well, at least I know ASV's is somewhere between 2,500 and 3,000, depending on how close we are to the meeting and how many people have just joined to get the cheaper registration and that sort of thing. So, but then there'd be overlap between the European one and
Yeah.
so
I just Googled how many virologists in the world, and the top hit was my blog post of 2013. How many viruses on Earth? Not viruses.
duh.
All right, let's do some. And back
Most
to the
of the virologists in the world are actually in the oceans.
Yes.
All right, so if we say there are, what would you think? Are there 10,000, 20,000? I wouldn't say there's less than 100,000 virologists.
Oh,
that's
absolutely.
less.
Right?
You're not counting clinical virology in a lab for the hospitals.
That's a good question. What is a virologist?
Right.
Is Dallin Dove a virologist?
is it alive?
Ask
Yeah.
Let's
Is it
ask
alive? Yes.
Alan, are you a virologist?
I think clinical virology in a hospital counts.
Sure. Yeah. Then that adds up.
Am I a virologist? You should ask me if I'm a virologist because.
Well,
I
have
would
you
say,
ever done a plaque assay?
Oh, is that the standard?
What's a plaque? No.
If that's
Might
the
be a lot of
standard,
people
we're,
doing research
we're
on
talking
viruses.
fewer and
Hydrated.
fewer.
No, I've never worked.
Okay.
I've never worked with viruses.
So the thing is that a young virologist will probably never have done a plaque assay.
Right.
So
Right.
that's not a gold standard, but
No.
No.
I've done a northern.
you could be something else and do northerns. Yeah.
And you probably don't want to include everybody who's used phage for cloning.
No, that's right because we've done that.
No, yes,
Right.
there are a lot of people. And what about people who are using viral vectors to deliver genes
Right.
to cells? That's all. They don't care.
Are they virologists?
And they just get the virus from someone else. No, they're not virologists. No.
No.
I mean,
They don't even know how many kinds of reproductive strategies viruses
so I don't
have.
know. I mean, if you go to a virus-focused meeting, like ASV is a broadly virus-focused meeting, you get 1,500 people.
Right.
If you go to the International Congress of Virology, that's bigger, right?
Could be.
Do they get? I've been, but I don't know how many people go to that. Does anyone know?
I don't know.
Never been.
5,000. We'll never know how many virologists there are. Would be
Wait a
interesting to
minute.
know.
They probably all listen to this.
It's very hard to quantify the virolome.
The virolome.
But if they are listening now, they should all write in, and it will cost you $1 on the crowdsourcing.
But they're not going to all be
Speak out.
listening. That's the problem.
They won't all be listening.
I mean, a guy came up to me after Cold Spring Harbor. This was the retrovirus meeting, and he said, "I'm now a listener after hearing it." I said, "Well, where were you before?"
Exactly.
But there are lots of virologists who don't know about it. Is it possible to quantify any profession in the world? Do we know how many MDs there are in the
Yeah,
world?
we do. Actually,
Yes,
We
you
do
can
know
you
that.
can quantify a licensed profession.
correct.
That's easy.
Oh, so then let's require virologists to have a license.
Have licenses.
Oh, yeah.
And
the
they have
numbers
to pay
will go down then.
the
Pay
license
$1 to
fee.
TWiV to get the
Yes,
license.
that's exactly right. Pay $1
Let's
to
start
TWiV,
issuing
That's what I
virology
said. That's
licenses.
just what I said before.
and
but
we will
that's-
give you a license.
That's will not.
Speaking of virology meetings,
So would that be a license to ill?
license to ill.
Ooh,
Very good.
nice.
Held every 3 years, the International Double-Stranded RNA Symposium is the premier conference for virologists interested in the biology of double-stranded RNA viruses. The 2018 symposium will be held in Houffalize in the beautiful Ardennes region of Belgium. The conference venue is close to Brussels, which provides easy access for European and international travelers. The symposium offers scientists from around the world the opportunity to meet and hear the most up-to-date advances on a broad area of topics in double-stranded RNA virology. The organizing committee have selected an international group of dynamic speakers to present plenary talks on topics including virus structure, diversity of double-stranded RNA viruses, evolution, epidemiology, using and abusing host pathways, immunity, pathogenesis, molecular and cellular virology, and applied double-stranded RNA virology. Students, postdocs, and scientists in the field of double-stranded RNA virology are encouraged to present their recent findings at the symposium. There are a significant number of travel grants available, in particular for attendees from developing countries. This year's program will also include TWiV with a special guest. Early bird registration ends May 31st. Link in the show notes.
I should go to that meeting because double-stranded RNA viruses are the ones that infect protozoans.
They do.
And there are numbers of protozoans that have pathogenesis factors dependent upon the presence or absence of double-stranded RNA viruses. So it should be an interesting meeting from lots of perspectives.
Yeah, I'm sure there are those virologists represented.
Yep.
How many parasitologists are there in the world?
Far less than there are virologists, that's for sure. Far less. Or should I say far fewer?
I was going to say.
A lot of them are working in somebody else's lab.
Yeah, maybe. You mean like...
Yes. Being parasites.
Arg.
Very good. Well, if anyone knows how many virologists there are in the world, let us know.
I tell you, we're going to fill up a jar, right, with marbles, and each one will be a virologist, and then they have to tell me how many marbles.
I know how you could find out. You could have a meeting and say, whoever comes will get a $1 million research grant.
Right.
And then we wouldn't know how to get the money, but we'd have all the virologists there.
You'd only get the gullible virologist.
Kathy, can you take that first follow-up email, please?
Sure. Sunny writes, here's a link to the pointer I think Kathy was talking about in TWiV episode 494. It definitely is a great tool to have when presenting. Keep on Twixing. And she gave a link to the Logitech Spotlight presentation remote. And that is it. It's really cool.
So what was special about this? Remind us.
So you have to watch the video, but basically, or even just go on the page and then you'll see a click. So where he's pointing to the Matterhorn. So Matterhorn is bright and then the rest of the slide has been grayed out while he's pointing to the Matterhorn.
Oh.
And how do they do that?
It's just some button you push on the remote.
Magic.
So, but okay. I think I want to wait for version 2.0 because I would want to be able to make the circle of brightness be smaller and point to the very top of the Matterhorn. So
I'm like a highlighter.
Hold out. Yeah.
In the strictest sense of that word.
The problem is it's $130, right? And I tend to leave these places.
For giving a talk at a university, they should give you one as a present.
Oh, yeah, they're going to do that. They won't even pay our salaries.
That's true.
I did get a laser pointer once
really?
at a place where I gave a seminar, but it was in Atlanta. And it was when I was thinking that, for once, I'll go to a baseball game and I'll have an overnight hotel and I'll pay for it with my honorarium. Only they gave me a laser pointer
instead.
Kathy, I never knew you were a baseball fan.
Yeah, I don't go to many games here, but it looks like I have 2 lined up in Baltimore in the near future.
Neat. Oh, Camden Yards is a very friendly place to go to.
I love it.
Yeah, it's a good place.
Cool.
Alan, can you take the next one?
Sure. Steve writes, here's an article and provides a link. Dry ice is certainly the method of choice. This is talking about the cold chain for Ebola virus vaccine. It's easy to handle safely. The procedures for air shipment are well defined. It's easy to produce on demand from ambient temperature compressed gas with little machinery. Doesn't require fragile or expensive containers like dewars. And it's not too cold, as is liquid nitrogen. And it won't carbonate vaccine or bananas that are in sealed containers. Those huge vaccine shipment boxes you see pictures of are probably mostly dry ice and, very importantly, terrific insulation. Not to minimize the many practical difficulties likely to be encountered in rural DRC, but maintaining the cold chain is just one among many.
Yep.
Good point. Yeah, just the way restriction enzymes are delivered to the lab.
Yep.
So you can make dry ice pretty easily.
Oh, sure. Yeah.
Easy.
Just take a CO2 cylinder with a regulator on it and squeeze, basically. And the CO2 coming out and expanding rapidly cools and freezes.
Neat.
Right.
You can get a cheap attachment for a CO2 cylinder that does exactly that.
All right, Dickson, we should try it later, okay?
We should.
Yeah.
Rich, can you take the next one?
Wink writes, Dear TWiV professors, thanks for the great Ebola update. As is customary, you began with a conversation about epidemiology. Missing from your discussion and all other modern reviews of Ebola is the originally reported mechanism for viral amplification in a human community. One reference below, and he gives a link. This was by reuse of needles in medical facilities. This practice may still be going on. There is no data to the contrary. It may be that the subject doesn't come up because it is too painful or embarrassing to think about, or hopefully the practice is merely historical. However, this was Peter Piot's initial finding. I think it at least deserves mention. Wink from Atlanta. So the link is to an article about the 1976 Ebola outbreak in Zaire, where there was a significant amount of transmission that had to do with the reuse of needles actually they were administering malaria medication. I forget exactly what it was, but they were reusing syringes on people who had Ebola, and it spread it around. That was not good. I would hope that, in particular, after the last epidemic and sort of instituting a lot of better upgraded public health measures, that much of that is taken care of. And
also in the context of the AIDS pandemic.
For sure.
Absolutely.
Yeah, needles were a big deal then.
Totally.
Yeah.
And hepatitis.
And of course, hepatitis C in Egypt was spread to many millions they were giving drugs for schistosomiasis, I think.
Yeah.
So that's a good question, whether I would guess it may still be happening in certain areas, but we don't know.
That's right.
It's a good point.
Yeah.
Duly noted, as we would say. All right, Ted writes, hello, TWiVerati. I wanted to follow up on last week's discussion of the paper from Heinz Feldman's lab, right? I know Jeremy provided email input that you included in the show, but it seems he mixed up Mayinga and Makona in his response. And I feel the need to clarify. Great. Good job, Jeremy. My main criticism of the paper, now I'm thinking back, he probably did mix it up.
Yeah, there was an issue there.
Because Mayinga is an older isolate that they used as a control, and he didn't like that, but that wasn't the point. The point were the two Makona isolates. All right, now Ted wants to tell us about that. My main criticism has to do with the viral isolates that were used in the study, which were generated via tissue culture passage, which I think you would always have to do, right? You always have to grow virus in cell culture. Especially Ebola virus. Great work from the labs of John Connor and Jim Cunningham has demonstrated that passage in culture often results in mutations in the fusion loop of Ebola glycoprotein. Perturbation of the fusion loop appears to have a similar biochemical consequence as the A82V changed, which is the subject of all of this. Increased propensity for the glycoprotein to initiate the series of conformational changes leading to membrane fusion. Perturbing the fusion loop appears to provide a greater membrane fusion enhancement than A82V. This is important because all the early isolates, A82 isolates used in this study, contain a mutation in their fusion loop. While it's unclear what the genotype of the other viral stalks are, if they match the NCBI accession number, they harbor A82V but no fusion loop alterations. Thus, it is possible that the early isolates were observed to have enhanced virulence because their fusion loop mutations enhance fusion more than A82V does. But I will pause there and note, it's a good point. I did send this to the first author of the paper, but she did not respond. Maybe she's on holiday. Maybe she's in monkey room. I don't know. But I would think, so they said in the paper that they had sequenced all the relevant areas. I don't know if they sequenced the whole glycoprotein coding region or what. I would think if they saw other changes, they would have mentioned them, but that's a good point. Okay. So point taken. And he continues, overall, this raises an important point of using isolates to test the effects of specific mutations. Using this approach, it's difficult to control for the genetic backbone of the viruses being compared. Engineering the mutation directly into a relevant virus backbone using reverse genetics and ensuring sequence fidelity of the recovered viral stocks is a better approach for direct comparisons. All right, let me stop there. It may not be a better comparison. So you're going to have to grow it in cell culture anyway.
Right.
It could be that one passage does this, right? You just don't know, in which case it's going to be hard to sort out. And the thing is, when you introduce A82V into an early Makona background, there may be other changes in late Makona that are relevant as well. You're not going to see those. So all you're going to look at is A82V. Now, that may be your goal, but and it should be done, but it is not the, quote, better approach necessarily. I mean, I think you have to start with clinical isolates, but if the passaging is an issue, I agree that's problematic too. Anyway, this paper also overlooks the epidemiological data. A82V was derived early during the epidemic, became widespread, and nearly displaced A82, was associated with increased mortality and higher viremia, the former statistically significant, the latter not. This could be due to genetic drift and founder effects, but all other published evidence, epidemiology, location of the mutation at the receptor binding interface, and biochemical changes to GP function indicates selection. I should note that this evidence includes in vitro replication studies with recombinant Ebola virus. It provides a link to a Journal of Virology paper. And this is all we discussed last time, whether it's a founder effect or actual selection. Right. We just don't know. And in fact, we will never know because we can't infect people. And as he says in a moment, the animal models are surrogates at best. Okay, so he continues. I also have a quibble with the animal model used. I believe the mouse model is largely irrelevant for testing the effects of A82V, as no effect of A82V was observed in murine cells. Additionally, while the gold standard proxy for human infection, the macaque Ebola model, is a classic example of monkeys exaggerate. This pathogenicity model results in 100% macaque fatality with a time to death of about 7 days. In contrast, the case fatality rate in humans is 40 to 80%, and the time to death is around 20 days. Because of these differences in comparison to human infection, it's possible that this pathogenesis model may not be suitable to detect subtle effects on virulence, but in my view, this is all the more reason that very carefully controlled experimentation be done to address the question. Lastly, it is possible that mutations that arose during the Makona outbreak facilitate transmission, which was not assessed in this study and may be prohibitively expensive to ever test.
Yes.
Well, that's the problem because this is the best you can do unless there's another model that no one's looked at yet that might be better, right? What was I just reading? Oh, yes, so mice are not a good model for herpes simplex infection, but guinea pigs are better. Who would have thought? So, getting close to humans, primates, not necessarily better. So
right.
Maybe you need to try some other models.
Right.
This paper, however, does suggest that further investigation is required. We couldn't agree more. We would like to see the isolates used in this paper compared using similar cell culture systems as performed by Stefan Becker's group to see if the robust in vitro phenotype can be recapitulated. Not to beat a dead horse, but a reverse genetics approach with non-human primates or other relevant pathogenesis model is needed to directly assess the role of A82V in vivo. Just remember, A82 does not exist on its own. It exists in a background of an entire genome. So, it's very, having done some of the first reverse genetics, I understand it's very appealing, but it's not always the best way. Finally, I felt the need to feed Vincent's pedantism. Was I pedantic? The title of the paper states that Makona mutations do not alter pathogenicity. Pathogenicity is a black and white characteristic. With a case fatality rate of 40%, it is clear that Ebola-Makona is pathogenic. Virulence is rather what the authors were testing in the study. Keep up the great work. Ted Deal. P.S. Sorry for the verbosity. And Ted is, of course, a postdoc in Jeremy Lubin's lab, who's actually William Deal but prefers Ted. Pathogenicity is a black and white characteristic. Some people use pathogenicity and virulence interchangeably. I know. I noticed that, and it's not correct normally. But yes, virulence is a quantification.
And by the way, in my Twitter stream, there was an extensive discussion of replies to me, and I guess Vincent was not on this, but very basically the same points covered by Nathan Grubaugh. Let's see who else was involved in this. Jonathan Ball. They were the main folks. Bob Cross. So just shout outs to all of them who essentially reiterated all of these same points, and we came to essentially the same agreement that this this is something that needs to be sorted out, and the people in the field need to get together and figure out exactly what it is they're testing and what their results mean.
So Jonathan Ball had written in 2016 saying it'd be useless to use monkeys to do these experiments.
Right.
But then we won't know an answer.
We wouldn't know anything.
Okay, because we cannot answer it in people. And anything else is an association. You do all the animal modeling you want. You do all the reverse genetics you want. In the end, it's all an association. I'd really like to see if this mutation crops up again in this recent outbreak, and why it's doing that. So, well, anyway, I think the Feldman group needs to answer some of these issues. And as I said, I emailed them, but they didn't answer. So they weren't part of that Twitter stream, most likely.
No, I don't think so.
Go ahead.
I would even like to know what started this recent Ebola outbreak. That's the $64 million.
Maybe they'll
question.
find out. What are the candidates, Dickson?
Bats.
Can't. Yeah, contact with bushmeat.
Bushmeat. Yeah.
Bushmeat.
Kathy.
I was just going to be a little bit more pedantic, because also part of that Twitter stream was the use of the term reverse genetics, and it's not necessarily that it's a jokey thing. It's that it's not unambiguous. Reverse genetics means different things to geneticists.
Yes.
To people in virology, it seems to be making an infectious clone of your virus. Why not just say infectious clone?
That's what we called
an infectious clone approach with non-human primates is needed to directly assess the role of A82B.
Hey, man.
Yeah, it's an unclear term. That's the complaint.
When we published our 1981 paper, we used infectious clone. And when Palese published his 15 years later, he started using reverse genetics. I don't know why. And people think it's nice and it works, but it doesn't. I agree. It doesn't work. And you can't say it on TWiV. Yeah, you can say what you want. It's fine. All right. Thank you, Ted. I saw Jeremy at Cold Spring Harbor. We did. And he said we had a nice discussion.
He's incognito, right?
Yeah.
He has a beard.
Huge. He looks like Chuck Darwin now.
Chuck Darwin.
Okay. Let's give a brief outbreak on this Ebola. No, let's give a brief update.
Update on the outbreak.
Ebola outbreak.
That's right.
There's a nice article in SIDRAP. As you know, there have been a couple of cases now in Bandaka. So they are tracking contacts. Health officials are tracking contacts of two patients who were taken out of an Ebola isolation facility in Bandaka, an event that may have exposed dozens of people to the virus. They just came and took them out to bring them to a prayer meeting.
Lord.
And then they died. They were vomiting and died hours after the prayer service. They were too sick to walk. Oh my gosh. Isn't that terrible?
Yep.
So
their relatives picked them up on motorbikes and drove them to a prayer meeting. Oh boy.
So they could have infected quite a few people, right?
That's right.
So the numbers at the moment,
I don't know how much contact there is in this prayer service, but one was dead and the other was dying afterwards. So
if they were unable to walk, they were symptomatic, so they were shedding. That's the key here. So as of May 23rd, there are 52 cases, 31 confirmed, 13 probable, 8 suspected. And some of the earlier ones that were suspected have been negative. But there are 3 new cases, 2 from Bikoro, 1 from Iboko. And so the vaccine, of course, is being readied for use. I don't know when they're going to do it.
I thought I heard that they started on Monday.
Yeah. And there's a very nice FAQ over at WHO, which explains different aspects. They're going to do ring vaccination, of course, around each laboratory-confirmed patient, regardless of the location. And contacts are defined as individuals who in the last 21 days lived in the same household, were visited by the patient after they developed symptoms, or visited or were in close physical contact. So those people at the prayer group should be vaccinated, I would think, right?
I would think so, yes.
The ring is not necessarily a contiguous geographic area, but captures a social network, right? That's right. Each experience suggests that each ring may be composed of an average of 150 people. You talk about the vaccination team, who's going to receive the vaccine. Is participation in ring vaccination voluntary? Pregnant and lactating women will not be given the vaccine.
And speaking of voluntary, people are referring to these patients as having escaped from the hospital.
Yeah.
And I don't know what the context is here at all. Is this a locked ward like you would have for a psych ward, or is this people leaving against medical advice and the term escape is just being tossed around? I don't know.
Yeah.
I get something like the latter. I can't imagine that it's, a locked ward or anything like that, but they're leaving against medical advice. I think that's my interpretation. And there's there's one of these articles, I forget whether it was the WHO FAQ or what, describes that there's a lot of superstition involved.
Sure.
In this disease, and going to prayer meetings is for some people an appropriate thing to do. So
right.
Yeah,
I just wanted to.
I think it's fine to go to a prayer meeting, but if you're sick with Ebola, you shouldn't.
No, you probably shouldn't,
because you're just going to harm a lot of other people, right?
Well, that's one of the things the teams that are doing the vaccination include. Anthropologists, sociologists who, and they're sort of the front line here to help educate people as to what's going on.
Yeah.
Right. And people who are from the area who are social mobilizers. I mean, they seem to be working with the local community to try and bring everybody on board the concept. So it's not a bunch of folks coming from out of town and telling everybody what to do.
Right. There's a whole social science of trusted messengers.
Yes.
And there, at least with HIV, the idea is to try and use clergy as the trusted messengers.
Right.
I just wanted to fill in just a little more technical detail on the vaccine, because you guys went over that really quickly last time. Even though we've done it before, there's a TWiV 349. We discussed the vaccination campaign at the end of the last Ebola outbreak. That used this same vaccine, but as was said on the last show, it's a recombinant vaccine in vesicular stomatitis virus, or VSV. Right. But I just wanted to, flesh that out a little bit more. Both vesicular stomatitis virus is an arthropod-borne virus of cattle, livestock, but mostly cattle, and the vector in this case is sandflies, and it causes in them a disease that is manifested by blistering in the mouth, and it's important, among other things, because it looks a lot like foot and mouth disease, which is a much more devastating infection, and so people have to be able to distinguish this. It can infect humans and cause a flu-like illness. That's a rare event. The most important point I wanted to make is that both of these are in the order, both Ebola and VSV are in the order mononegavirales, that is single-stranded negative sense viruses, though they're in different families, VSV being a rhabdovirus, same family as rabies, and Ebola being in the filoviruses. But they have the same, a very similar genetic and physical structure. So there's a genetic content and a gene order that is similar in the two viruses, and they both make a nucleocapsid that's in a wrapped in a matrix and then an envelope, and sticking out of the envelope, embedded in the envelope and sticking out of it, is the glycoprotein that's used for attachment. And so this recombinant virus takes the glycoprotein from Ebola and swaps out the VSV glycoprotein and replaces it with the Ebola glycoprotein. So it's not like two wildly different viruses that you're making a recombinant from, which of course you can do as well. But this is two similar viruses, and it's a live or a replicating recombinant vectored virus. So the VSV can actually replicate and use the Ebola virus glycoprotein in its replication cycle, and you can actually grow this as a virus itself, and then use that as a vaccine. There's only one other or two other licensed recombinant vaccines used in humans, both based on the flavivirus, yellow fever virus backbone. We've discussed those before, one for Japanese encephalitis virus and the other for dengue. So these recombinant vectored vaccines are not very common. They're coming into human use more and more, and I'm interested to see how this works out. It's a much more common phenomenon in veterinary vaccines. There are many more recombinant vectored vaccines.
Okay. Next, there happens to be at the same time an outbreak of Nipah in India. Nipah virus. 12 people have died, and this is a paramyxovirus. Bats. I mean, the original Nipah outbreak was a transmission from bats to pigs and then from pigs to humans.
Right.
More recently, they've stopped that mode of transmission. More recently, bats contaminating date palm sap, which people collect at night, and then they drink it and they get infected because the bats go in it in the night. And the solution to that was to just cover the containers that you collect the sap. In this case, this seems to have started with... they think a bat in the home of the people who got sick. They're testing the bat, and then it's spread among the family. And there's an article in SIDRAP about this, which discusses the Coalition for Epidemic Preparedness Innovations. CEPI is announced a $25 million collaboration with two companies to develop a vaccine, which is great, but should have started a long time ago because this virus was discovered, or at least it was identified in 1998, and lots of people have continued to die. But what I find very interesting here is that they say this vaccine, they're going to do correlative immunity studies at a BSL-4 facility at Galveston National Laboratory. And that's like the very beginning before you can make a vaccine, right?
That's figuring out how to make a vaccine.
Yeah. Now, Nipah, another paramyxovirus, highly lethal, again, from bats to horses to people.
Do you mean Hendra?
Did I say Nipah? Yeah,
you
Hendra.
Said
Nipah, Hendra. Hendra is another paramyxovirus. The original outbreak in Hendra, Australia, which is, I believe, a suburb of Brisbane. And there, bats to racehorses to people. And so a vaccine was developed which is given to racehorses, and that stops the transmission to people. So you don't need to immunize all the people because there's very few cases, but you can immunize the horses. Now, in this case, you can't immunize date palm sap or pigs or the bats, so you have to immunize people. But I think it'll be a while before they make this vaccine, nevertheless.
I've been digging around trying to figure out what this vaccine is, and I've come up with all kinds of stuff.
Which one? Which one are you talking about?
The Nipah vaccine. Well, it's confusing. Yeah, the Nipah vaccine is what I've been chasing around, but and I've erased most of it now because I thought this article, the CEPI article that's linked in this about the vaccine used in Australia. If you dig into that, you find the quote that the vaccine component is just a single protein from the virus. It's the glycoprotein. So it's a subunit vaccine.
That's right.
There's a bunch of other vaccines out there that are other recombinants of one sort or another, but I gather that this vaccine is just the subunit vaccine, the glycoprotein.
When I was there visiting Geelong, which is in Australia, has the BSL-4 lab, where the Hendra vaccine was developed, Lin Fa Wang was involved in that. It's amazing. So the BSL-4 there can accommodate 50 large animals in the BSL-4, like cows or horses. I couldn't go in because it's active, but so that's where they did challenge tests in horses of the Hendra vaccine, and they needed to know where the virus was replicating, and they had to get the horse brains out, right? So they had to sacrifice the animals, And he said the only way they could do it was with a chainsaw.
Oh,
Can you imagine?
chainsaw and a BSL-4.
Rich.
That generates a lot of aerosol.
Yeah.
It's a chainsaw virus.
Oh, that's good. Anyway. Okay. Lots of things going on.
Yeah.
So,
And they all happened after my course ended, because I always say at the beginning of the course, my virology course, something's going to happen during this course, and it happened the week after. Although the Ebola has been going on for a little bit longer, so we just didn't know about it.
right.
I just wanted to mention the CEPI thing that I hadn't really heard about until this year, because they're sort of not sponsoring, but they're a good part of the organization of one of the ASV 2018 satellite meetings on Saturday about vaccines. And so this was founded to speed the development and fund new vaccine candidates. It's supported by governments of Norway, Germany, India, and Japan, and also by the Bill and Melinda Gates Foundation, the Wellcome Trust, and the World Economic Forum.
Right.
Great. Get going. I wonder how long it'll take.
And also, we had 2 TWiVs in the past, at least, about Nipah. The one that Vincent made when he visited Lin Fa Wang, TWiV 296 from August of 2014, and then one TWiV 58 from 2009.
Wow.
Joshua Stillman talking about passive antibody therapy for Nipah in ferrets.
I don't even remember that one.
I thought you remembered all of these things.
Joshua Stillman, really?
I copied that right out of the show notes. I couldn't have made it
Go
up.
back to that one.
I don't even remember him. Yeah, Nipah virus. You go. Yeah. Oh, he's the emergency doc here. Remember him?
Josh Stillman.
Oh, right.
Yeah, he came here right here in the office, and you knew him.
I know him very well because he participated in our course in parasitic diseases.
Yeah, that's right. I remember. He looked very tired. Now I remember. Yes.
He's always tired.
He had a cup of coffee.
That's right.
Probably always does. Okay, we have 2 papers, so we need to move because Alan has to leave in 1 hour,
That's right.
and they're both very cool. First one is a PNAS paper called "Symbiotic Polydnavirus and Venom Reveal Parasitoid to its Hyperparasitoids." Now, that's not going to make any sense to you, but once we tell you about it, it will make perfect sense.
I have a naive question. Why did you say polydna when it's poly-DNA virus?
Because people say polydnavirus.
Yeah, they say,
But
I try and pronounce things properly,
understand
although
that.
I don't always get it right.
if you say poly-DNA, at least you know what kind of virus it is.
Polydna.
Right.
Okay, you got it?
I've got
Yep.
it.
And the authors, the first 2 authors are co-first authors, Feng Zhu and Antonino Cusumano. And the last author is Eric Polman. And this comes from Wageningen University in the Netherlands, the Netherlands Institute of Ecology,
It's a good
the
place.
Vrije Universiteit Amsterdam, and the Max Planck Institute for Chemical Ecology.
And this, by the way, was sent to us by our friend Christopher Mendoza, who
Yes.
started the podcast Cast 9 and had a polydnavirus episode on that, and has his own interest in polydnaviruses. He dug this up and couldn't resist sending it to us. Good find.
Right. Polydnaviruses are features of parasitoid wasps. Parasitoid wasps, they inject their eggs into other insects, and along with them, they inject polydnaviruses. The viruses disrupt the immune responses of the hosts, so their progeny are not taken care of, so they can survive. And this is an amazing symbiosis. The polydnaviruses are old; they're over 100 million years old, and they are part of the genome of the parasitoid wasp.
So this is an old infection that got integrated into the genome and then repurposed by evolution to help the wasp in its parasitic lifestyle.
And we've talked about this quite a bit.
Oh, yes.
Yeah.
This is an arc for sure. We like this. We have to have the fellow from Georgia. What's his name?
Mike Strand.
We have to have him on sometime and have him.
Yeah,
Mike single or double strand?
Polydna.
And as a matter of fact,
Polydna,
there was a postdoc who worked for him for a long time called Galen Burke, who who's also now got a faculty position at Georgia. Have them on at the same time or something like that. Get them on at an ASV meeting if they show up,
Now, so the additional level of this is you've got the parasitoids that are laying their eggs in usually a lepidopteran caterpillar.
Right.
And then they, according to the old rhyme, have smaller fleas to bite them. So they have hyperparasitoids that lay their eggs in the larvae or the pupae of the parasitoids.
Right.
So after the parasitoid wasp lays its eggs in the caterpillar, and those the larvae eat their way out of the caterpillar, often killing it, then those pupate on the outside of the caterpillar. A hyperparasitoid comes along, lays its eggs in that pupa, and then that hatches out of the wasp pupa that parasitized the caterpillar.
And that's bad for the parasitoid, obviously. It can kill, it says up to 55% mortality to the parasitoid.
Which is important in agriculture because these parasitoid wasps of various types are used extensively as biological control for caterpillars that eat commercially important plants.
And Wageningen University in Holland is one of the leading agricultural schools in the world.
Okay, so I wanted to start this by saying that if you want to draw this as a line, there's a cabbage at the left end in this set of experiments. And in fact, they use two different varieties of cabbage, one to propagate their caterpillars and one when they're actually doing their experiments with their caterpillars. And so then there's the caterpillar that's then, and it's called the large cabbage white butterfly. So it's a caterpillar that goes on large cabbages. And then there's this parasitoid that Alan described, and then it's parasitized by the hyperparasitoid.
Right.
So kind of moving left to right, you have a whole chain, and they use the word multitrophic in this, a multitrophic chain phrase in this paper, and I had to kind of figure out what they meant by multitrophic.
So the cabbage is a producer. It takes sunlight and turns it into food, and then everything else is a consumer. You have the caterpillar is a primary consumer, then the parasitoid, I guess, is a secondary consumer, and the hyperparasitoid is tertiary.
Right.
So this paper wants to know, how do the hyperparasitoids know to put their eggs into a parasitoid-infected caterpillar? Right.
Yeah, and importantly in this discussion, as Alan said, the caterpillars ultimately die from this, but in this particular case and in other cases, up until the point when at least when the hyperparasitoid shows up on the scene, the caterpillar is actually still alive. So you can have caterpillars that are so far advanced in this that the larvae have emerged and are spinning cocoons on the outside of the caterpillar. This is alien in spades, right?
Yes.
And the caterpillar is still alive and eating the leaf, and that's important.
Right.
They are so screwed, these caterpillars.
But in eating the leaves, if they are parasitized, when they eat, they regurgitate. And there's some saliva in what they regurgitate. And if they're parasitized, their regurgitate is different colored from if they're not parasitized.
Right.
And this all will elicit volatiles. Okay.
From the plant.
Go on.
From the plant.
From the plant.
Who, by the way, is the real victim here.
Right.
Plant?
Yes.
The plant's getting eaten.
Yes.
Right.
So it doesn't want to get eaten.
So, yes, that's the key here. Through all this chain, in the end, the volatiles are coming from the plant that then attracts the hyperparasitoid.
That's a pseudopheromone that a plant produces to attract an insect. That's remarkable. The first example I ever heard of was the tobacco hornworm that was eating tobacco plants. And tobacco plants that are eaten will secrete a pheromone that attracts a parasitoid wasp to interfere with the moth's ability to feed.
In that case, it makes
hyperbiology.
Sense. Right, it makes sense because the parasitoid is going to eat what's eating the tobacco plant. In this case,
I know.
It seems like it's not really advantageous because the plant volatiles are going to attract a hyperparasitoid that is going to kill or damage the population of the parasitoid that was otherwise going to be helpful to the plant.
Yeah.
Mm-hmm. The cabbage hasn't had time to counteract
it's
the hyperparasitoid because I think that's where we are in this arms race.
Right.
So this old volatile business in viruses. We've talked about a long time ago, actually, an aphid virus that the aphid delivers it to the plant. The plants make volatiles that attract more aphids. They come and they pick up the virus, and it tastes badly because of the infection, and immediately the aphids leave and they bring the virus somewhere else. Does anyone remember that paper? It was ages ago. It's very cool stuff. And then, of course, there was the beetle that holds something, the puppet master.
Yes, that's right.
It gets paralyzed until the things are developed. I can't remember what exactly it was. So here the question is, what is doing this? Because the wasp puts in eggs, the virus, and venom.
Right.
And so they have a cool system where they have 2 jars, and they have plants in the 2 jars. And then
excuse me, let me back up for a minute here. The issue is that the hyperparasitoid, they knew going into this, can distinguish between a caterpillar that has been parasitized and one that cannot.
That's correct.
So it'll go to the caterpillar that has been parasitized. And so the question becomes, how does he know how to do this? She. Okay.
So they have a Y maze system where they have 2 jars with plants in them, connected by tubes in a Y configuration, and they allow the either infected or uninfected caterpillars to feed on these plants, and they take them out along with the feces, just have a plant, and then they put a wasp at the other end and they see where it goes.
Give it the Pepsi challenge, yes.
The Pepsi
exactly.
challenge. And so they take and they do combinations of eggs and venom and virus, which they get separately. And it's the venom, so the virus alone is enough to cause attraction of the wasp, but the venom makes it even more. It makes it work better.
And if you have access to PNAS, this is paywalled, but if you look at Figure 1, expect to kind of settle in and spend, I don't know, 20, 30 minutes staring at this figure, figuring out all the hieroglyphics and graphics that they've used to describe what's a very, very comprehensive set of experiments here.
And it is the regurgitate. And as Kathy said, the different colored regurgitate compared, in fact, parasitized as opposed to unparasitized.
Would you call that the hurlome?
The hurlome.
The hurlome, yes.
And in fact, so the saliva that's coming out of these caterpillars, one of the sources is the labial glands. And these are known to secrete a variety of compounds that can digest plant tissue. They remove them from the caterpillars. And if you remove them, that gets rid of this ability to induce the right volatiles on the plant after you've been parasitized and hyperparasitized, right? So changes in the gland are crucial for this chain of interactions that allows the hyperparasitoids to find the parasitoid larvae. It's all starting in the saliva glands.
Right.
So then they take it one step further. They say, what's in this salivary gland, and they do transcriptomic analysis. They do RNA-seq, and they ask what's different between salivary glands of unparasitized and parasitized caterpillars. And they find genes that go up and down, and they say 2 of them are interesting. Beta-glucosidase and glucose dehydrogenase are differentially expressed, and there's some evidence that these might be involved in the induction of volatiles from the plant. That's the next step here. How does this work? And they say, we don't know how it works, and we have to keep working on it. So it's the virus alone can do this. And so
there are also virus genes expressed in the salivary glands.
Yeah, that's right.
So those could be having an effect directly.
Probably, right? And that's where we end.
So does the virus replicate in the caterpillar cells?
If this polytenovirus works the same as the others, first of all, the genes that are delivered are encapsulated in a virus capsid protein, but the genes themselves are not viral genes. They're immunomodulatory genes that are amplified out of the wasp host. And those, this is, in the polytenoviruses that I know about, those encapsulated genes do penetrate the caterpillar cells and in fact even integrate into the genome of the caterpillar cells, I believe, or at least are expressed. Now, you don't make more virus, you make protein products that are immunomodulatory and help suppress the caterpillar's immune response. So I was thinking about this. It is possible that some of this virus, which is injected along with the eggs, can infect glands can infect cells of the salivary gland and make some of these proteins. And whether or not those proteins actually affect the regurgitate and therefore affect the plant volatiles and impact on the hyperparasitoids' attraction to the infected caterpillar, that remains to be seen.
So their RNA-seq revealed some polydnavirus gene expression in the salivary, but the virus doesn't replicate. There's no production of new virus particles. Right. Yeah.
Right.
So that's the key now.
So the polydnavirus is initially injected along with the original parasitism in order to suppress the caterpillar's immune response, but apparently that has the side effect of somehow affecting that caterpillar's regurgitate, which then influences the production of plant volatiles, which cues the hyperparasitoid that, this caterpillar's infected, and I can hyperparasitize them. My God, I'm thinking Russian dolls the whole time.
Yes.
Well, I would, think about the sequence of evolutionary events that led to this, right? I mean, did the regurgitate, was it different in the beginning before the hyperparasitoid? And if so, what was the function of that? I mean, really cool.
Yeah, I mean, it seems reasonable that it could have happened either way, but I kind of see it happening as this parasitoid relationship developed. The wasp is parasitizing the caterpillar, and then another wasp develops the ability to parasitize the parasitoid, and as part of that, the selective pressure would be finding your host, and these volatiles coming from the plant, which happen to be different in the infected caterpillar case, are driving it there, and it happens to be the viral, contribution to all this that's allowing that to happen.
I just
Go ahead. I was just going to say, this paper has kind of an odd structure, I think, and a whole lot of experiment description that I think would ordinarily be in the results is in the materials and methods.
Yes, right.
And so
that's right.
This hyperparasitoid is the most common hyperparasitoid of the parasitoid in the Netherlands. And it says that it is found waiting next to parasitized caterpillars until the parasitoid larvae leave the caterpillar to spin their cocoon. So, so they're just sitting there, and they can be parasitized. The parasitoids can be hyperparasitized up until 2 days after the cocoons have formed, and that ordinarily it sounds as if the parasitoids could be attracted by some odorants also, but the plant volatiles are produced in much higher amounts than what you could get from the parasitoid itself, because they say the silk cocoons of the parasitoid emit low quantities of volatiles and are not strongly attractive to the hyperparasitoid. And so the whole timing of this is important too, that because they talked earlier about the hyperparasitoids might arrive too early to plants when they can't discriminate between the volatiles. And so then that sounds like that's why they just sit there and wait.
They wait. How long do they have to wait, do you know?
Well, they didn't say that. I don't know.
Until it's ripe.
So the larvae emerge. Then they get them. Then they get them.
But how much too soon did they get there, I think is what you were asking.
Yeah, I just want to know if it's, what is I don't know. I don't know offhand the whole developmental timing. Rich, do you know from the moment the wasp injects eggs, how long does it take for them to hatch?
No, I don't know. I found, by the way, a pretty good YouTube video that we could consider putting into the show notes here on the hyperparasitoidism, and
I like
so it shows all this happening, and it's clear that the caterpillar you say Helen the caterpillar is screwed,
Yes.
but is still alive while all of
Yes.
this is happening.
That's
He's
even
moving
worse.
around. Yeah.
And by the way, they seem to indicate about 14 days for
the whole thing.
The whole thing.
Yeah,
So when does the hyperparasitoid
that's
come along?
Right. So 14 days from the laying of the eggs by the parasitoid in the caterpillar till the emergence of the larvae chewing their way out of the caterpillar to spin their cocoons.
Mm-hmm.
At which time the hyperparasitoid
At which
is
point time the hyperparasitoid, possibly sitting there for some time, comes in and injects and does the same thing to the parasitoid.
Amazing, huh?
Amazing.
I'm just waiting for somebody to discover a hyperparasitoid.
Yeah.
It
Maybe.
exists, I'm sure.
I want to just point out that this assay is called a Y-tube olfactometer assay.
Which I looked up. There's a bunch of different images of that.
Yep.
One other line out of the meth is the parasitoid is gregarious
Yes.
and lays up to 35 eggs per parasitism event.
We would call it fecund, right?
Yes.
Fecund.
Okay.
Fecund. Not gregarious. That's weird.
Now, gregarious
methods
means
are
that they like
really-
to hang out together.
The methods are really long because they put all of the description in there. Yeah.
Yeah.
It's weird, but
So- And I wrote to the senior author, Eric Pullman, to ask him for a diagram, but it was because I had just read the introduction and I hadn't looked forward to figure 1 yet where there is a figure. So that's the figure that he sent me. But when I was looking at stuff on his site, they have a kind of it looks like a Popular Science Dutch TV or type Dutch podcast kind of thing that tells the whole story in Dutch. But the opening of it is really cool. There's a 20-second ad, and then there's an opening of the podcast that's just really cool graphics. So I think it might be fun to include that link also.
Sure.
And if anybody is Dutch, they can
Right.
watch that.
So
I
they-
was also amazed that they could operate on these caterpillars and remove their salivary glands,
and
and then
do the
Yes,
assay,
they'll keep going on as if nothing was happening.
keep chewing on the
these
leaves.
caterpillars, well, they're accustomed to being eaten from the inside by these parasitoids. So these are pretty tough caterpillars.
They keep their appetite. They go to the leaves, start chewing them, regurgitate. Now
Can they still go to the opera?
that's
Right.
I don't know.
We'll always have parents.
This is the kind of story
You can't
that
keep a good moth down.
this is the kind of
Ooh.
story that people like,
They
right?
do, sure.
Because not only is it viruses, but it's going
complicated.
inside
Biology.
and
It's biology.
complicated,
And it's
work
creepy
out of it. It's
and-
creepy,
yeah.
exactly.
That's right. The body snatchers.
No, not body snatchers, but alien.
Alien.
Developing
That
inside
too.
the idea of developing inside another organism, even though many animals do that. Right?
Yeah.
We do that.
when it's put there by a wasp, a parasitoid.
Vincent, all
I know
human
we do.
beings-
Of course, I know we do.
That's
that.
our own species.
I understand
but when
that.
put there by another species.
Yes,
You
another
know,
species is pretty weird.
I totally
But
agree.
you know what? They say this is probably really common.
It
We
is.
just don't know it. Okay.
Ichneumon wasps are found
Cool,
everywhere.
cool paper. Now, for a little more mundane, we have a cell-hosted micropaper, which I liked. It's different. A sialylated voltage-dependent calcium channel binds hemagglutinin and mediates influenza A virus entry into mammalian cells. Not quite mediate. So I would say mediate brings it in, but it doesn't really bring it in. Facilitates. Exactly. Thank you. Facilitates. Yoichiro Fujioka is the first author, and Yusuke Oba is the last author. This comes from Hokkaido University, the National Institute of Infectious Diseases, and the Marine Biological Laboratory in Woods Hole. Wow. It's a book about influenza A viruses, which, as everyone knows, cause the respiratory disease known as influenza. They bind, to get into cells, they bind sialic acid receptors. Sialic acid is a little sugar that's attached at the very end of a sugar chain, which is attached to proteins. And so influenza virus is attached to this sugar. The hemagglutinin binds the sialic acid in a nice little pocket at the tip of the HA. It's brought into the cell by endocytosis. Now, conventional
Sialic acid's on a lot of things.
sialic acid's on
Right.
things.
No, that's
And
right.
people have, since I started working on flu as a grad student, people have always said, is there a specific protein that the virus binds to? But no one can ever identify one. And we know that if you take sialic acid off cells with, say, neuraminidase, then the virus can't infect. In fact, that was one of the earliest receptors identified because neuraminases are widespread. You can get them from Vibrios, for example, and other sources, and people treated cells with them and the flu wouldn't infect. Now, so in any case, the virus binds sialic acid, then they're taken up by endocytosis. And then that's where this story begins. Calcium, intracellular calcium, upregulates endocytosis. This is known in all sorts of cells, especially in neurons. It's really important for neurotransmission. And in fact, it's known that when you infect cells with influenza A virus, there's oscillation of cytosolic calcium. It goes up and down.
Right.
And-
So normally the cell's keeping the calcium out.
Yeah,
And it has these channels
that.
that'll let calcium in and
Yep.
signal certain things.
Yeah, you want to keep the cytosolic calcium pretty low, and there are transporters of all sorts that do that, and then there are channels that will let it in when you need it. And these channels, they're called calcium channels, like voltage-dependent calcium channels. Put a little voltage on and they open up. Inositol triphosphate-gated calcium channels, and then ryanodine receptors. These all let calcium in. And these guys, these investigators focused on voltage-dependent calcium channels because a number of reasons. They're at the plasma membrane. Their function can be modified by sialylation, the addition of sialic acid, and verapamil. Verapamil. It's a calcium channel blocker. It was shown in 1984 to block influenza A virus replication. So these individuals said, let's look at voltage-dependent calcium channels and see if they play some role.
These calcium channels are also in the right place. They're in the respiratory epithelium.
Right.
Some of them are, yes. Not all of them are. Yes, some of them
Not
are.
all.
So the general family of voltage-dependent calcium channels are in various places, but they're kind of zooming in, looking at circumstantial evidence where might the specific receptor for flu be, and they came to these voltage-dependent calcium channels.
Now, the advantage here, the cool part is that there are lots of blockers of these channels, of these voltage-dependent calcium channel blockers like verapamil, diltiazem, amlodipine, okay?
I am not.
And others. And they tried all these, and they inhibit virus infection, virus production in a dose-dependent manner. And they're not toxic to cells. And they
and by the way, the reason there are so many of these calcium channel blockers is because they treat high blood pressure.
Right.
Exactly.
So there's been a tremendous amount of research and development done on this particular class of drugs, and you have a bunch of them sitting around that you can treat cells with, and the cells survive, and you get a very specific effect.
Yes, and as we know, not to diverge into high blood pressure, but my wife did a long project on it at Merck. Many people can reduce their high blood pressure by limiting their salt consumption, but others cannot, in which case you have to use these things. But other people like to eat salt, and so they take their verapamil, Anyway, they focused on diltiazem. How do you say it? Diltiazem? Diltiazem.
Diltiazem is how I'd probably pronounce it, but I don't know.
So they focus on that. That's the most potent. It doesn't completely block infection, but it does a pretty good job. And they look in a variety of cells. So they look at some where you can get virus production and it inhibits production of virus by plaque assay. But in other cells that are not productive, they use an immunofluorescence assay. And they say in multiple cell types, you can see an inhibition of infection by diltiazem. Okay? So that suggests that these voltage-dependent calcium channels somehow are playing a role. Right. Now, there are lots of these. There are different kinds that are produced in different tissues, and they have numbers like 1.1, 1.3, and 1.4.
Right.
Turns out 1.2 is in the right place, so they focus on that one. They knock it down by siRNA, and the knockdown reduces virus infection. So it looks like this 1.2 voltage-gated channel is important.
That must have been a happy day.
Yeah.
Yeah.
I was wondering why they don't knock it out using CRISPR technology. Maybe it's essential.
I don't know.
Probably. Yeah, I would.
Or it was
imagine
quicker to do the initial experiment this way. I don't know.
Right. And they do this in multiple cells. So looks like this 1.2 calcium channel is important. They then say, how's this working? What's going on? So they, this, by the way, this voltage dependent calcium channel 1.2 has 24 transmembrane segments. You can see in the picture, and they're divided into segments 1, 2, 3, and 4. So that what they do is they produce these segments of the channel, and they say, will they bind virus? Simple binding assay, membrane binding assay. And they find that the number 4 segment preferentially binds. And this binding is inhibited by an enzyme that removes sialic acid. But it's not inhibited by a calcium chelator, the binding of the virus to segment 4 of this channel. So it needs sialic acid. And guess what? There are 2 sialic acid sites on segment 4. So they say, can the virus bind? Can the HA bind this? And they make a soluble form of the HA, which is secreted from cells, and they show that this can bind this the segment 4 of the channel, but not other parts of the channel, and that binding depends on sialic acid on the channel.
A competitive inhibitor.
So here's what we got. We got this voltage-gated calcium channel. The influenza virus can bind to it. It seems to bind a sialylated region of the channel, and that's important for infection. So then they make altered forms of the channel where the residues, the amino acids that are normally sialylated, are changed so that the protein can't be sialylated anymore. And they find that those don't work well as receptors, and the binding is reduced as a consequence of removing the sialic acid genetically as opposed to enzymatically before. And then they say they have cells where they deplete the channel and they put in either wild type channels or the sialic acid altered channels, and only the wild types work well. So it all looks like the virus is binding to this channel at the sialic acid site on residue 4. All right, so this is all in cultured cells. What about in animals? So they look in mice where there are these channels in the respiratory tract. They can infect these mice with influenza virus, and they can treat them intranasally. You have to do that because if you put it in the blood, it doesn't get to the respiratory tract, these blockers, with diltiazem, either before or after infection. If you treat them before infection, it reduces virus yields pretty well. Not as good as a neuraminidase inhibitor like Tamiflu, but it does pretty well. If you treat them after infection, it's less good at blocking. But it does work, and it also it prevents mortality in the mice if you give them a higher dose. Again, it's not as good as the Tamiflu, but it does work.
Right.
And then finally, they do experiments in human respiratory epithelial cells, which they can grow into mimics of alveoli or the epithelial surface. And they show that this drug, diltiazem, can decrease influenza virus infection. So that's basically the story. So the idea here is that the virus binds to this channel. That binding allows calcium to come into the cell, which stimulates endocytosis, and then the virus gets in by being endocytosed, probably on a different receptor on the cell surface. So they haven't shown that this channel is endocytosed at all. I guess it could be, right?
So
Yeah,
could be.
why do you think it has to be a different receptor?
Yeah.
Because in their picture in the front, that's what they show.
Well, okay.
Yeah, look at the graphical abstract. They show the virus activating the channel, but actually being endocytosed on another protein, other sialylated proteins in the endosome going in. So, I mean, I tried to find this, whether this particular channel is endocytosed, and I couldn't find it. I mean, it's well known that calcium through these channels stimulates endocytosis. of other proteins. And so but it could be, I suppose, that the channel itself comes in, but they didn't show that in this paper. They just showed
that may just not be known.
Yeah. But it does stimulate infection and makes sense because we know that endocytosis requires calcium. And the cool part here is that the virus is activating the channel to let calcium in, right?
What's the particle to PFU ratio typically on influenza A?
It is a couple hundred.
So you're going to have a bunch of particles kicking around that are not going to maybe not get into the cell.
Mm-hmm. Remember, we did some defective flu papers.
Right.
It can vary.
It can vary.
There are lots. Yeah, there can be a lot of.
But there's going to be a lot of them that aren't getting in, and they might be the ones that bind to the calcium channel, and then the others get endocytosed on anything else that's got sialic acid tag on it.
Yeah, these could be facilitators.
Right.
Yeah, we're not talking about something that's black and white 100% here. Where all of these effects that are described in the paper are, as you say, stimulatory or facilitating. Okay, it's perfectly logical that the virus is binding and perhaps internalizing elsewhere, and that this phenomenon is a stimulatory effect.
Right.
So I have a question. The last sentence of the first paragraph in the discussion, they say the findings are expected to lead to the development of a novel approach to inhibiting influenza infection by targeting the HA calcium channel interaction. Such therapeutics appear to be effective for a range of serotypes and tolerant to drug-resistant strains. But that second sentence, I don't know that we have data for that.
I didn't see any drug-resistant strains in this paper, did you?
Well, or that it would be tolerant. Yeah. So anyway, then I was trying to think, okay, this is going to be, this is an antiviral. So trying to think like a virus, how is the virus going to evade this antiviral?
What is the target of? Is it the channel or the HA that's the target? It's not clear, right? They just say we're going to target the interaction.
Right.
Right.
I don't think you want to use calcium channel blockers to treat flu, right?
Well,
isn't that
people are already on them, and that doesn't seem to have stopped the spread of flu.
I wonder.
Millions and millions of people take these drugs.
Well, actually, that would be an interesting epidemiological study, right?
Yeah. Are they at lower risk of the flu?
Yeah, right.
Right.
I mean, one drawback of that is that they're going to be you'd have to pick your controls carefully.
Yeah.
Right.
Yeah. I suppose you could design a channel blocker that did not actually block the channel, but you couldn't. That's the whole point of its inhibition.
Right, channel blocker. Yeah.
It's got to be a channel blocker. Yeah.
Mm-hmm.
And if you targeted the sialic acid binding site, then that's just
it's only a channel blocker if you don't want your children to watch.
Yes.
So they're very excited about this, using it as an antiviral, which is okay. But I think the mechanistic part is really interesting. That
Right.
virus is actually facilitating its own uptake, right? That's pretty cool by allowing calcium to get in. So again, it's not whether the virus gets in via these channels, we don't know. But as they say, this is probably not the only protein involved in uptake. Yes.
Are you saying that the influenza virus sends the wrong message to the cell?
I don't know if it's wrong. I mean,
no cell wants to be infected, but the cell can't avoid it once the signal is initiated, right?
Apparently.
Right. The signal gets the cell endocytosing, which increases its susceptibility to the virus.
Any other viruses work this way, Vincent?
Good question. Calcium uptake? Well, yes, I would say yes, because endocytosis depends on calcium uptake. So all the viruses that are taken up by endocytosis, and that, by the way, that there are lots of different kinds of endocytic pathways, right? There are clathrin mediated and clathrin independent. They apparently all require calcium. So I would guess that a lot of viruses. So now the question is, would these inhibit other viruses, right?
Yes.
Good question. Have to go get some verapamil. We can check it out.
Yeah.
not verapamil, the other one. I always like that word, verapamil.
Yeah, me too. Remind me again what verapamil was therapeutically used for, though I thought it was tuberculosis, but it was
high blood pressure, right?
Yeah,
Alan?
but there's an infectious disease it's used for too, and I forget.
Really?
Yeah.
It's a treatment of high blood pressure, chest pain from supraventricular tachycardia, migraines, cluster headaches. It doesn't say tuberculosis.
Okay. It's in my head. So
It's in your head?
Somewhere. I'll be looking for that reference.
Yes.
That would be the migraine.
There was just a new migraine drug approved,
Yes.
right?
That's right. That's right.
What is that?
I don't know, but it was a big deal.
Apparently.
Oh, yeah. On our TWiP show, Daniel Griffin mentioned it because his
he
daughter
lamented
suffered.
it being $600, right?
Yeah, his daughter suffers from migraine, so he's going to probably be using that.
Yeah. All right. That is it for our papers. Let's move on to some email. The first one is from James. No, let's read. Dr. Condit, you should read this. It's all about you.
Not all, but yeah. James writes, "Hi, TWiV team. On TWiV 451, you had a conversation regarding whether or not, quote, all science is wrong, end quote. During this discussion, Dr. Condit mentioned the Cochrane collaboration, which performs meta-analysis of published studies. This reminded me of a resource aimed at performing meta-analysis of RNA-seq data. It has a great name, the LAIR. Here's the link to the paper, gives a link, and the website gives to the link to the website. I should mention that as a scientist myself, I agree that not all science is wrong. And I think meta-analysis tools like this empower the scientific community to double-check findings, especially for big data that is hard to look at without proper training. If nothing else, the homepage of the website has a cute picture of a bear on it, which is indeed true. On a more personal note, I wanted to say a quick hello to Dr. Condit, who was on my thesis committee at the University of Florida for about 2 years before his retirement. I'll never forget an amazing talk he gave to the incoming graduate students on the do's and don'ts of being a scientist. After that, I was desperate to soak up as much of his wisdom as possible and was thrilled Dr. Condit agreed to be on my committee despite being at the end of his tenure at UF. Also, Dr. Condit and the rest of the TWiV gang, I want to thank you for inspiring me to remain passionate about doing and loving science. I recently started my postdoc at the Fred Hutch in Seattle, and there have been quite a few growing pains, read unfunded fellowship applications, which have occasionally put a damper on my belief of I have what it takes. That said, I recently picked TWiV back up after almost a year-long hiatus, and it has been a lifesaver. Really, just hearing one of you say, "Isn't that cool?" in response to some finding in a paper you discuss makes me believe that I never have to lose that same feeling of amazement. Anyways, just a big thanks to you all. I'm excited to catch back up with TWiV and haven't been wasting my time 4 episodes alone today. Thanks all again, James Thomas. P.S. Included in the growing pains mentioned above are all of this rain and lack of sun. Phew, we're not in Florida anymore.
Yep. Fred Hutch.
So, James, I've been thinking about this. First of all, thank you. I think a teacher's greatest reward is the acknowledgement that he or she had an impact on somebody's life. And so I really appreciate that. At precisely your time in my first postdoc, I was ready to quit. Changes like this are really hard. Getting into a new system, being in a new place, it's one of the most difficult things a person can do. And in particular, getting into a new system. Lots of times stuff doesn't work. I had, not a positive result for like 9 months into my first postdoc. And I started to think, maybe this is the wrong thing for me.
Rich, what would you have done if you had quit?
Well, that was it. I had no
Golf.
plan B. Okay. There was not even biotech. There was no backup plan. Nowadays, there are many more plan Bs. But one of the things I wanted to, obviously I got through to the end. Okay. You guys Kathy and the rest of you must have experienced all of this same kind of stuff. There are downtimes.
Oh yeah.
All right. And James, I know you have what it takes. You've got the fundamental motivation, the fire in the belly that showed up from day one. Not only that, but you got the best training that you can get. The Swanson lab just absolutely can't be beat. So hang in there. It'll be fine.
Hey, Rich, maybe you would have joined your friend and made some sausage. He's rich now, I think, right?
Yeah, he can buy and sell us all. Yeah.
So Astel, is that his name?
Adels.
So I go into a store somewhere, and it was full of one of those big box stores, Sam's or BJ or something. It's full of Adels sausage.
That's right.
I took a picture. I sent it to Rich. Hey, your buddy here is cleaning up. He's got a picture of him outside a pub in England on his website. And
can buy and sell us all.
Funny.
I try to warn people when they're finishing their PhD and starting their postdoc that you're going to go from being at the top of your game, knowing how to do all the experiments in your lab and where everything is and how to find anything in any other lab that you need, to going to a new lab where the undergraduate dishwasher knows more than you do about where stuff is, or and but that you have what it takes to figure it out, and you will.
I was so scared going into my postdoc. I'm going to David Baltimore. I said, oh, crap, I'm not going to know anybody. Nobody's going to talk to me. I won't know any. I was just completely frightened. And I got there, and he said, go clone polio, and he left. Okay. And he said, talk to Al. And I went to Al, and he said, come back next week. Oh, okay. And I came back, and I kept going back. And I said, look, I got to do this. And eventually, you get over it. You got to push. If it's what you want to do, you have to push through it, right? Yep. As the lady said the other night on TWiV, I asked her, what advice do you have for people? Because she just is 6 months into her assistant professorship. She said, you just have to keep your passion.
That's right.
Don't lose it, because that drives everything. Alan, can you take the next one?
Sure. Jenny writes, hi, TWiVsters. In episode 493, Rich shared his experience of an FDA advisory committee meeting. After mentioning the committee included a patient representative, Rich said he wondered about the process for selecting that rep. I participate in FDA's patient representative program and can share some info. FDA was an early adopter of incorporating patient experiences into decision-making, largely as a result of HIV/AIDS activism. Today, the program trains and provides representatives for FDA advisory committees and other review decision assignments. You can read more about the program here, provides a link. Individuals can apply to the program, and FDA looks for several qualifications as detailed at the link. Representatives are compensated for their time and must complete regular training as well as financial disclosures. When preparing to serve on an advisory committee, representatives, like other voting members of these committees, must undergo a detailed conflict of interest clearance process to ensure they have no financial conflicts. Representatives are appointed for multi-year terms with no guarantee they will have the opportunity to serve on committees. Our experiences of specific diseases may never be relevant to drug or device reviews. Those with experience related to diseases that come up in reviews more frequently, some cancers come to mind, may have more opportunities. In my experience, the FDA takes patient perspectives very seriously. The training is excellent, and so are FDA staff. Participating in the program has been a very positive experience for me personally. Cool. So they've just got a population of these people who they've recruited and trained, and when a panel is being convened for a particular drug, they tap into their patients for that condition, their representative, and bring somebody in.
Mm-hmm.
So and if you look at this website she gave, it's got criteria for becoming an FDA patient representative, and one of the bullet points is personal experience with the disease, either as a patient or primary caregiver. The disease in this case was smallpox.
Right.
And this was a relatively young woman, so I can't believe that she had experience directly with smallpox, but maybe some other runaway poxvirus infection, one of these circumstances with a vaccinia infection, vaccine complication or something like that.
look at this. They must undergo a detailed conflict of interest clearance process. It's even more rigorous than if you wanted to be president of the U.S.
I told you that to be a special government employee, I filled out 17 forms. Okay. And they know more about my finances now than I do. Okay.
So it has to be an odd number so you could
exactly
make a vote. Dickson, can you take the last one?
Chris writes, Dear TWiV team, I was intrigued by some of your comments about the mode of action of tecovirimat. It sounded to me like you thought that an inhibitor of spreading of viruses from one cell to another was unusual and wasn't likely to be particularly effective because it wouldn't affect the production of virus particles within an infected cell. This is not so unusual, however, after all. Fusion inhibitors of HIV target the entry of virus into a new host cell. The protease inhibitors of HIV and HCV target the maturation of viruses, leading to the production of non-infectious virus. And the neuraminidase inhibitors of influenza viruses target the release of progeny viruses from infected cells. Hence, inhibitors of fusion, protease, and neuraminidase inhibit the spread of virus to new host cells. Provided one stage of the replication cycle is targeted, that should inhibit overall production of virus. Targeting more than one stage of the replication cycle is likely to have advantages, of course, particularly in terms of inhibiting the development of drug resistance. I assume that because since pox viruses are double-stranded DNA viruses, mutations are likely to arise much less frequently than a virus such as HIV, where the use of multiple drugs targeting multiple stages of the replication cycle is standard practice. Thanks for all your informative and entertaining podcasts. Best wishes, Chris.
Right on the money. Yeah, those are good examples.
Mm-hmm.
And so, Rich, you would agree that it's not so unusual to stop the spread?
I would now. Now that Chris is tuned me in.
Right.
Yeah, I mean, he was right. And the initial reaction to tecovirimat was this isn't going to work because it just inhibits spread. And people still talk about it that way. But Chris makes an excellent point. There's plenty of other drugs in the HIV realm that effectively do the same thing. So it's not so uncommon.
All right. We have a final email from Tom who said we don't have to include this in the podcast, but he's talking about subscription rates for science, which we were musing about. It's about $100 a year, he says, to the bi-monthly Science News. But that's not science, right?
Right.
No.
So you have to click on the link.
Yeah.
And then it gives you that a regular rate is
I see. Yeah.
165 for print, 125 for digital.
I think it's worth every penny.
And then there's student postdoc rates, teacher rates, retired rates.
I sit at home and I get the thing delivered to my home and I have coffee and I eat my oranges and grapefruit and I read the news about science.
TWiV is cheaper.
Is it? What?
And by the
now
way,
you tell me.
anybody could get the science advocate rate.
Mm-hmm.
Individuals who are not scientists or engineers but believe in the importance of science for our society, and that's a $50. Well, $65 is the digital rate, $95 is the regular print rate, and they're running a special right now.
Neat.
It is.
That's good.
So that's I think that may be a recent addition.
So the only thing is, there are so many sources of good research. So
yes,
correct.
That's why you have to have a different strategy. If you're really interested in the papers, not the front matter, right? Because here you can get a nice smattering of different
front matter is fun to read too.
Yeah, out of your field, I think.
That's right.
But I like to do RSS feeds, and that's how I look for TWiV papers.
It does include the Science News Online too.
Yeah,
right.
Yeah.
So,
and he also subscribes. So Tom is from Austin, who we will meet presumably next month. He also subscribes to Scientific American over 55 years ago. Wow.
Ron was right.
It's a good. I like the Atlantic.
Mm-hmm.
I don't subscribe to it, but I love the writing, and I like the New Yorker writing.
My wife subscribes to the New Yorker, so
I can't read it anymore. I have to read science articles. Not enough time. All right, let's do Alan's pick because he's got to leave.
Okay, yeah, sometime in the next few minutes. So my pick is someplace I went about a month ago. My daughter had spring break and we didn't have anything planned, so just took her down to the D.C. area, and we went to a couple of museums. The Smithsonian, of course, everybody's heard about that. But this one is pretty close to where I grew up. It is the National Cryptologic Museum. And
Hard to
I'm
find.
not making a joke when I say it's a little hard to find.
Right. Sorry. Didn't mean to step on your line.
It is actually somewhat poorly labeled. People have commented on this, but it is off a major highway. So once you get to it, it's a small museum. Takes maybe a couple of hours to go through if you're taking your time and appreciating it. And they just have a neat collection of displays and artifacts of the history of code breaking and of the NSA, the National Security Agency, which is right there in Fort Meade, Maryland. So this Cryptologic Museum is an outreach effort of theirs, which I guess was a little odd for NSA to do, but they did. Yeah, no, they're not really public-facing kind of organization, sort of the opposite. But you can go in there. You can see NSA headquarters from there with big satellite dishes on the roof and everything. And it's just a really cool set of displays and explanations of the science behind codes and ciphers and how they've been made and broken. And they have probably the world's largest collection of Enigma machines, including one you can actually touch. Which may be the only place in the world where the general public can go in and just tap on the keys of an Enigma machine. I thought that was kind of cool. So just a nifty place if you happen to be in the area and you're looking for entertainment for a couple of hours at the museum.
Did you go to the Vigilance Museum next door and see the airplanes?
No, I didn't catch that. It was pouring rain when we were there. But
I have to do an arc on this one. I was in Kazakhstan last week at a meeting in Astana, and the meeting center had rooms, and each one was named after a famous person, and my talk was in the Alan Turing room.
Oh.
Cool.
That's very cool.
Yeah, it was really neat.
I'm sorry the Brits killed him.
Yeah.
Very sorry. Can you imagine where computing would be today? It's terrible.
No, of course not.
Terrible.
No,
Thank you, Alan. Do you want to stick around? Or do you want to go right now?
I'll stick around. I'm all right.
Rich, what do you have for us?
Just something I found probably on a social media somewhere. It's an article in The Verge titled "Stock Photos of Scientists Reveal That Science is Mostly About Staring."
I love the opening photo.
That's great. Everybody looking at that chicken rooster.
Chicken. This is written by somebody who was
deciding which came first.
Yes, you got the eggs there.
Right. So this was written by somebody who was trying to find a photo of a scientist to describe what they did for a living, and was Googling through various photos of scientists and found that all of them are, or the vast majority are people who are holding things up and looking at them.
Right.
That's the conclusion.
Sure.
There's one great one here of somebody with a bunch of eye protection on looking at a piece of dry ice.
Yeah.
Exactly.
And at any rate, this started a Twitter thing that's hashtag bad stock photos of my job. And if you go to that and look at all those, it really gets off and it's great. This is the one I really like. There's a picture of a guy in full protection regalia with a hood and goggles and gloves and everything else, pouring blue fluid from an Erlenmeyer into a test tube in a rack.
You bet.
And is talking freehand pour very dangerous blue liquid, VDBL, from a beaker into a tiny tube in a rack you hold with your other hand. Mandatory eye and respiratory protection because VDBL. Pretty good.
Rich,
It's hilarious.
I'm sure you've been involved in this, and I know have too, and many people. I mean, there are listeners all over that are scientists that when they do PR for the university, they want to come to your lab to take a picture.
Yeah.
What's the first thing they want you to do?
Oh, they have you look through a microscope.
Exactly. Mm-hmm. And it doesn't matter what microscope, and it doesn't matter.
You either have to be looking through a microscope or pipetting.
Those two things. That's
my wife and I have a standing joke about that.
Anytime
Anytime there's science news, they'll show somebody in a lab pipetting.
No question.
B-roll. B-roll.
Yeah, B-roll.
Listen, once a reporter from AC 360 came here, right? Anderson Cooper 360. there was a norovirus outbreak on a cruise ship here. She's like, I want to come up and I want to see norovirus. I said, well,
Exactly.
come up. I'll tell you about it. So I'm talking about it. I even have my little models. She said, but I want to see it. I want to see you inactivate it. So I got my student to come over and spray the countertop with bleach. And I said, there, that's it. I said, but my producer wants to see it.
You should have had a ray gun.
So you can't. It's too small. I don't have an electron microscope. And so they did all this filming.
I know.
And then it never ran because
I know.
she didn't see it.
It's horrible. It's just horrible. Or they have these, the football games, they have that during halftime. Each college gets a chance to sort of tout themselves, right? And they show people dancing or they show people running in sports. And then they show, of course, the invariable laboratory scene, white lab coat, microscope, test tubes. It's all the same.
And at the end, she left, and I said, she said, you're not very happy with me, are you? I said, well, you don't seem to understand. She said, well, science is not my beat. It's usually crime.
Right.
But she said, I did get an A.
Show me your gun.
She said, I got an A in biology in college.
Arg.
We used to watch in movies or TV clips or things like that for whether there were Kim Wipes boxes anywhere on the scene. And if there were not, then we knew it wasn't really
Right.
a
That's a
valid
good
lab.
That's a good
I like to say you need to look for the duct tape.
duct tape.
I was out at Irvine with Rich, right? And you weren't with me, Rich, but Bert took us to his lab, and Ray Ortega was with us. And what did he do? He went right up to the cell culture hood, Ray, and he starts videoing the lady pipetting.
Sure.
He said, I need it for B-roll.
Or there's an ad for a breath mint or something like this. It kills 99% of the microbes in your mouth, and they show pictures of planaria or paramecium.
Great. Dickson, do you have a pick?
Of course I do. This is a fantastic journey into a cave in New Zealand to see the famous glowworms, which are not really worms. They're larvae of gnats. There's little dipterans that hatch out of these glowworm cocoons eventually. But in order to grow, they have to capture things. They're carnivorous. And they secrete these strands of mucus with little beads on them, but they contain luminous bacteria. All right? So you go into these caves and they start to flicker. And they're doing this to attract their prey to get inside, to get trapped on these strands of mucus.
There are no staring scientists in this video.
There are none. In fact, I would have loved to have seen someone do a rolled up scientist in one of these little, these are not small glowworms. These are big.
Yeah.
They need the reverse camera because we're staring at it.
Yeah.
Well,
Yes.
there you go. There you go. But and they're synchronous almost. They're flashing almost at the same rate. So it's quite
Very nice.
quite wonderful. I wish next time I'm in New Zealand, I'm going to go down and look for them.
Kathy, what do you have?
Well, I have two picks. The first is that there's a big exhibition at the Smithsonian for several years, and celebrating the opening of that was an event on Monday morning. The exhibit is called Outbreak, and they live streamed it, but also a video of it has been archived at a link on Facebook. And so I put that link in there. The first person who talked, Stefano Bertuzzi from ASM was there, and he introduced Stacy Solch-Cherry, president of ASV. She gave a short little talk. And then Mary Woolley from Research America, and Vincent, you've talked about that before. And then Tony Fauci, director of NIAID, and the CDC director of the National Center for Immunization and Respiratory Diseases, Nancy Messonnier. I didn't hear how her name was pronounced. Anyway, so they all talked about various aspects of vaccines. It was in particular because of the 100th anniversary of the 1918 influenza and so forth. And Mary Woolley presented data from a collaboration between ASM and Research America, asking a bunch of questions about vaccines and things. And so that was interesting. And while the live stream was going on, I got the email from Research America giving the link to her talk. So I put that link there.
Cool.
So that's my first pick. So you can check out this exhibit at the Smithsonian over the next several years. There's going to be some aspect of it at the ASM Microbe in Atlanta in a couple of weeks. And then, Vincent, you have a comment in here, too.
Yeah, we're going to do a TWiV at some point in the museum. ASM is sponsoring that.
If you go to the outbreak exhibit on the right day, Niles will show you through it.
Mhm.
Oh,
Is he a
cool.
He's a
He's a
docent?
docent at the museum.
That's cool. Very good.
Yeah, they mentioned that they trained, I don't know, like 60 docent volunteers. So it's a large group, unlike other exhibits at the Smithsonian.
Yeah, the communications committee of ASM got a fellow, so they I think it was funded by both the Smithsonian and ASM, and I was involved in getting that. And that fellow is a scientist who helped train the docents who were not scientists to be able to explain the exhibit. It's really
Cool.
nice. Yeah.
So, and then my second link relates to attending summer meetings, including ASM Microbe 2018, which is in just a couple of weeks in Atlanta, as I said. So my first link is to the Dynamic Ecology blog site, and they have a whole bunch of links there about logistics, like traveling to meetings while you're breastfeeding, how to meet people and network, how to give a good talk or a poster, how to answer tough questions, perfecting your elevator pitch. So it's just a great resource for a bunch of links. And then something more specific for American Society for Virology. Last year, we asked 2 people to write a survival guide for the American Society for Virology meeting so that you can get the most out of it, particularly how to deal with the fact that there are concurrent sessions and you can't be everywhere at once. And so just relax, go see some of it. And that was written by Matt Evans and Victoria Meliopoulos. And that link is in here too. It's up on the ASV website and also the meeting website. So that's it.
All right.
Cool.
My pick is ICTV Online, the International Committee on the Taxonomy of Viruses. This was prompted by a visit. I visited University of Alabama, Birmingham, and my first visit of the day on Tuesday was Elliot Lefkowitz, who is the data secretary for the ICTV, and he showed me their website, which has been revamped. And if you know ICTV, they categorize all the viruses. They have meetings of different committees. They listen to proposals for categorization, and they have a taxonomy list here on the website, which is great. You can find your virus there. And the cool thing is they used to publish this huge book every few years that was very expensive, but now it's no longer published. It's all online. So the 10th version of the taxonomy is online. They show you all the proposals, the history of classification of different viruses. It's a really cool resource.
So how many viruses are there, Vincent?
So if you go, I asked him that, and he showed me the list. So these are the ones that have been isolated and characterized.
Yeah,
okay, but there's slightly over 4,000, and there's a historical list of them, starting with a few hundred. Some years it actually goes down as they reclassify and goes back up. And then he said, of course, now they will take only sequence in the absence of infectious virus, but it has to be complete sequence, and you have to have a lot of information about it.
Okay, another academic question then. When HIV, which is a virus, infects an individual and begins to exhibit changes due to its random mutation rate, how different is it from the original virus before it's considered a new species?
Well, they define that here, and that the percent change that you would have to go through to make it a new species, and you can go find that there. Go to the ICTV online.
Thank you. I will do that.
It's there. It's right there. They have a definition. I mean, it's kind of an arbitrary limit, but how much change would make a new species? Totally. ICTV online. It's very cool. It's free.
Okay.
Check it out.
I'm bookmarking it. Yeah. And also, they have the logo of the ASV meeting in Maryland because they are truly sponsoring one of the satellite meetings.
Oh, good.
So they're entirely doing that. It's all about a world of viruses, exploding diversity, and its impact on classification.
That's TWiV 495 microbe.tv/TWiV. Send your questions and comments, please, to TWiV@microbe.tv. If you'd like to support us, go to microbe.tv/contribute. You could give, for example, $1 a month. That would really help us out. And Dickson Despommier, thank you. You were at parasiteswithoutborders.com, Racaniello.org, thelivingriver.com. Thank you, Dickson.
It was a pleasure.
Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thank you. This is a lot of fun.
Rich Condit is Professor Emeritus, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Alan Dove is at turbidplaque.com and on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. We have a lot of titles today.
Boy, do we.
Well,
many of them are very good. Beam me in, Scotty. Too much of a good thing. I like Influenza keeps its ion channel 20. 20 is what?
It's the atomic number for calcium.
Oh.
I was trying to figure that. Well, yeah, at channel
It's
20. It's 1.2. It's
You have to think.
okay.
A channel? It's a channel.
Beyond the ion curtain.
It's
is
the
great.
ion channel. Yeah. Yeah, that's good.
Influenza lives in a gated community. It's funny. The very hungry hyperparasitoid is the very hungry caterpillar, right?
Yeah.
I still have that book.
The ion sleeps tonight. That's a good one.
Hey, Rich, which one do you like?
What's the long arm of the slaw?
The cabbage.
Cabbage. That's pretty cool. Can we take out the 20? Influenza keeps its ion channel.
Sure.
I don't know if it keeps is the right thing, though, right?
Yeah, sure.
You guys like that?
Yeah.
Kathy?
But then there's not a channel number.
Has to be a channel.
Oh, that's right. There has to be a number because it's TV, right?
Yeah.
Or
so if you want to put in parens after 20 calcium plus, that would be, sort of giving it away. But that would be fine with me.
It's an inside joke. It is.
It is.
Yeah, you could always play this as an Easter egg too.
Inside joke.
Do you
Could have a contest.
It's a good one.
See if any of the listeners understand the title.
It's an inside joke.
All right.
That's how you get inside.
Work with everyone. Influenza keeps its ion channel 20.
Okay.